Okay, thank you. And thank you for coming. So I'm going to walk you through a series of studies today that myself and many others have worked on over the past few years that make incremental steps towards understanding the role of the human cerebellum in cognition. I can't figure out how to advance my slides. I'm touching it. Okay, there we go. So while much of the neuroimaging community focuses on the cerebral cortex, we of course know that there's another brain beneath the cortex. The mouses. There we go. And the human cerebellum really is a powerful computational structure. It comprises about 80% of all the neurons in the brain, making up a total of the fifth of brain metabolism, and when flattened, it's estimated at about 80% of the total surface area of the cortex. And we, of course, know for a long time that the cerebellum has been involved in cognitive processes that extend well beyond motor function. So, for example, we know a lot from comparative anatomy that lateral hemispheres of the cerebellum really extended, increased in association with the association cortices. Sorry, the mouse isn't really working here. And we also know from transneuronal tracing studies that much of the association cortex projects transsynaptically to the cerebellum. And finally, we know from neuropsychology that many patients with cerebellar degeneration have impairments on a wide range of cognitive functions, from executive function, verbal working memory, to spatial cognition. And then finally, we know from task-based fMRI that the cerebellum is activated in response to a large number of cognitive tasks. And we know that the cerebellum is reliably activated by motor function, but more so than that, beyond representation of hand motor presses, saccades, action observation, the cerebellum is encoding a lot of information related to cognitive processes. And of course, one of the holy grail questions in my mind of cognitive neuroscience is really to understand the role of the cerebellum in cognition. And one of the reasons we talk about the cerebellum having a unique role is because of its relatively homogeneous microcircuitry. So no matter where you look in the cerebellum, the same structure is repeated throughout. And this gives rise to the idea of a uniform function that's applied across a wide array of tasks. And while much of the neuroimaging approach has largely focused on the cerebellum using task contrasts or gradients or even parcellations, we of course know there's another important structure missing from this picture, which is the cerebral cortex. And we know from work by Randy Buckners and many others that there is a nice functional correspondence between cortical networks and corresponding regions of the cerebellum. And this was shown using resting state functional connectivity, where each voxel of the cerebellum is tagged with the cortical network with which it's most functionally correlated. So again, you see those blue networks, this double representation, are mapped onto somatotopic representations of the cortex, and the pink and red are represented largely by frontoparietal and default mode networks of the cortex. But really what we're interested in doing is understanding cerebellar activation within the context of this cortical activation, because many different tasks may activate the cerebellum, but in order to really understand its unique role in cognition, we need to understand how its function relative to cortical contributions. And to do this, we need to build a model that establishes the true mapping between the cortex and the cerebellum. And then any deviations from this model we can consider as selective cerebellar function. And so to do this, we took a simple machine learning approach. We leveraged a whole bunch of different tasks that we collected in earlier experiment. So these represent a large number of tasks across a group of participants scanned over 4 different sessions. And in total, it constitutes about 5.5 hours of data. And so we trained a model by learning the mapping between the cerebral cortex and the cerebellum on one set of these data. And then we could take those model weights and generate novel predictions of the cerebellum using new cortical data. And we think this can put this question to a strong test. What is the cerebellum selectively doing in cognition? And so to do this, we trained a bunch of different models, each at different levels of granularity. So from coarse granularity, 7 different networks, up to many thousand different regions. And we did this for random tessellations of the cortex, but also we did this for many functional parcellations of the cortex, which I'm not showing here. And then we wanted to vary the sparsity of the model, so how many cortical inputs we were allowing in to predict what was going on in the cerebellum. And so to do this, we leveraged different regularization approaches. So we could take a ridge regression model, which allows for many cortical inputs to account for the variation in each cerebellar voxel. We could take a more sparse model, which allows for a few different cortical regions to influence each cerebellar voxel, and then the least or high sparse model, which is a winner-take-all. So essentially what we see with the Buchner cortico-cerebellar maps, where we allow for one region. And what we find is, on the y-axis, I'm showing the predictive accuracy, so how well these cortico-cerebellar models can predict each cerebellar voxel. And what we find is that models with low sparsity, so that allow for many cortical regions, perform best of all, followed then by intermediate sparsity, and then winner-take-all performs worst of all. And this demonstrates 2 important findings here, which is that a cortico-cerebellar model, the best one, is characterized by a distribution of cortical inputs, but then, rather than a very sparse model. But then, secondly, it also demonstrates that 60% of the variance in the cerebellum in this model is explained by what's going on in the cortex. And this is good or bad news, depending on the audience. So for cerebellar imaging, it means still that we can keep doing cerebellar imaging because at least 40% isn't explained, meaning that there are deviations from this model that are likely, the cerebellum is likely doing something unique in this 40%. And so we can take this best performing model now, and we can learn some interesting functional insights from this. So where we're interested in understanding the cortical weights or the cortical connectivity maps associated with different functional regions of the cerebellum. And so given the known somatotopy in the cerebellum, we can see here that regions that activate the left-hand cortex, or sorry, the right-hand motor cortex is encoding regions in the left cerebellum. Similarly, for the right cerebellum is encoded by regions in left motor cortex. And then you have this region in ocular motor vermis that is typically encoded by saccades, is then functionally connected with regions in frontal eye field, and then action observation regions, which are activated by watching people perform different actions, are encoded by regions of the frontal cortex associated with premotor, somatosensory motor regions. And then beyond these motor regions, largely what you see is a pattern of frontoparietal networks here that are encoding for each of these different cerebellar regions. And so when we can see that there's clearly a distribution of these cortical inputs across the cortex, and so we wanted to actually quantify this, to quantify the convergence between cortex and cerebellum. And so to do this, we could just take for each cerebellar voxel the percentage of the cortical surface area and calculate that. And then we can also measure the dispersion, so how far apart these different cortical regions are for each cerebellar voxel. And when we did this, I'm plotting the convergence from high to low, so high being red and low being blue. And what we can see for both of these different measures is that you have what sort of looks like an anterior to posterior gradient in a way, where you have high convergence in regions of the cerebellum that are functionally associated with frontoparietal networks, default mode networks, sort of more cognitive regions. And so the advantage now of having a falsifiable model like this is that we can now test functional theories of the cerebellum, and this was great work that was done by Ladd and Shoshani, who took this model, so took these connectivity weights and evaluated new cerebellar data. And one way in which she did this was she took 2 tasks, one a speeded motor task, which is known to both reliably activate the cerebellum, But it's also thought to be necessary for the coordination of rapid alternating movements, and this is often impaired in patients with cerebellar degeneration. And she took another task, a high-force task, which similarly activates both cortex and cerebellum, but doesn't seem to be impaired in the same set of patients. And now she was able to take this model and evaluate these cortical data to determine whether the cerebellum had a selective role in either one of these different tasks. And again, what I'm plotting here on the x-axis is the predicted activation, so how well the model predicted these cerebellar activity patterns, and on the y-axis is the observed cerebellar activation data. And if we look first at the regression line, again, we can see that this model, which was built on a separate set of participants, does pretty well at generalizing to this entirely new set of participants, new tasks, new scanner, new everything. Sorry, not the same scanner. But what we see are these characteristic deviations from the model. So these high speed and medium speed conditions are above the regression line. So these regions were underestimated by the model. So the connectivity model wasn't able to fully estimate or predict these two different conditions, suggesting that the cerebellum may be selectively recruited for speeded movements, which is really interesting. And we then wanted to test this, but now across different cognitive tasks. So in a different experiment, we set out to test participants across 5 unique tasks, so 5 unique domains that we've tested before in previous work. Different tasks, for example, a working memory Maedbh task, a semantic prediction task from the language domain. And then we had these 2 new tasks, a social prediction and an action prediction. And we varied difficulty across each of these different tasks. And I won't go into all of the tasks for time, but for the 2 new tasks, we recorded varsity soccer players here taking hours and hours of penalty shots against 16 different targets. And then on the bottom, we recorded again many hours of people performing social interactions, so hugging and shaking hands. And we showed participants these videos in the scanner, and we asked them to predict the next sequence. So they were asked, will the player kick to the left or to the right in the action observation? And then in the bottom, will the friends hug or shake hands? And maybe it's a bit difficult to see here, but difficulty was modulated based on how much information was shown in these videos to each participant. So an easy condition for the action observation, you can see the players sort of kicking the ball to the right of the goalkeeper, whereas it's more difficult to tell with the hard condition. And for the social condition, the man in this video has 2 arms outstretched, indicating he's going in for a hug, whereas in the other condition, it's a bit more difficult. So participants had to learn these tasks, and they got to be able to do these pretty well. And so we then took the exact same approach as I showed earlier, in which we took our connectivity model and predicted cerebellar activity patterns across these different tasks. And again, what we found was a pretty strong prediction in terms of the connectivity model was able to predict what the cerebellum was doing for the most part, but then we did have these classic deviations, and we saw this mostly for the prediction tasks, so for these dynamic action and social prediction tasks, we saw the cerebellum was underestimating what was happening in these 2. And when we look at the residuals of this model, again, we see that the action and social here are above the regression line. And interestingly, the social prediction task seems to be a little bit more selective than action prediction for a cerebellar involvement, which is interesting. And so we could, do a lot of speculation as to why the other tasks weren't selectively recruited by the cerebellum. These are tasks that reliably activate the cerebellum. There's a big history of each of these tasks, working memory in particular, and language, and I'm not saying the cerebellum is not involved in these tasks by any means. But one way in which I think we can move forward as a field, and maybe this is applicable generally in neuroimaging, but especially in cerebellar neuroimaging, is to really consider neuropsychology as well. And I think neuropsychology really complements neuroimaging approaches, because within the cerebellar field, there's lots of misalignment between neuropsychology and neuroimaging. You have many, many papers on tasks that activate the cerebellum reliably, especially within the cognitive domain. But then you also have papers from the clinical world that find a more mixed bag of results, or find no deficits or very subtle deficits in some of these same tasks. And I think neuroimaging alone really isn't sufficient to be able to get at the question of unique cerebellar role in cognition. And we need computational models of behavior. We need insights from clinicians to complement some of these approaches, and that's something that I think we should think about going forward with the cerebellum. And so just to wrap up, what I've introduced today is a novel way or novel approach to evaluating cerebellar function in cognition, providing some preliminary insight for selective involvement in dynamic action and social prediction tasks. And then, of course, in going forward, I think this symposium is about the lifespan and about developmental insights, which I haven't talked about. All these experiments were run on mature, adult, healthy brains. I think, of course, the cerebellum has been implicated in developmental disorders, as we'll soon hear. And we can really take these models now and evaluate them across the lifespan. I think that will be a really fruitful approach going forward. And thank you for your attention.
Hi, so if anyone has any questions, thank you, Maedbh, that was fabulous. I can pass the mic around.